Hello everyone, welcome to my channel Vani's Biology. Today we are exploring one of the most fascinating processes in microbiology, that is bacterial conjugation. This is the story of how bacteria share DNA and how scientists discover it using minimal medium and complete medium, and how it became a powerful tool for mapping gene. By the end, you will understand F factor, HFR strain, and even solve a genetic mapping problem with me. Let's start with basic. What is media? In microbiology, media are nutrient-rich substances used to support the growth and study of bacteria in a laboratory setting. Media is two types. One is minimal medium and another is complete medium. In minimal medium, contain only the essential carbon source and some salt and trace element. From minimal medium, the organism can synthesize all the other components for their growth and reproduction. If a bacterium can grow here, it means it can synthesize all the amino acid, nucleotide, vitamin, and the precursor for DNA and RNA. On the other hand, complete medium is nutrient rich. In complete medium, supplies vitamin and amino acid and all kind of substances. Those bacteria with biosynthetic mutation can grow here because everything is supplied externally. So let's talk about the stain. Two different type of stain. Those are grow in different medium. There are two type of stain. One is Auxotroph and another is Prototroph. The Prototroph stain that is wild type and that can synthesize all essential nutrient. And Prototroph need no nutritional supplement for their growth. But autotroph strain are unable to synthesize essential nutrient. Autotroph strain, that is mutant strain that cannot grow on minimal media. They require supplementation to survive. For example, E. coli strain with tryptophan, adenine and thymine genotype. And this thymine plus indicate this strain is wild type for that gene. That means thiamine gene is wild type. This strain can biosynthesize thiamine. But tryptophan and adenine, there is no plus sign. That indicate this strain is mutant for this gene. If there is minus sign is present instead of blank, that also indicate mutant strain for this gene. This strain cannot synthesize tryptophan and adenine on its own. This strain require tryptophan and adenine to be supplied in the growth medium. These strains grow on a two different medium. One is minimal medium and another is complete medium. This strain will not grow on minimal medium because it has mutant gene tryptophan and adenine. In minimal medium, lack added tryptophan and adenine. It only grow on complete medium in which supply all nutrients, or minimal medium that's supplemented with the amino acid tryptophan and the adenine. After that, 1946, Lederberg and Tatum discovered conjugation by this experiment, where they studied two E. coli strains that different in different nutritional requirement. Strain A and strain B both have the genotype methionine, biotin, threonine, leucine and thymine. In strain A, threonine, leucine and thymine have plus sign. This plus sign indicate that this strain is wild type for this gene, and methionine and biotin don't have any plus sign that indicate this strain is mutant for this gene. And the strain B, methionine and biotin have plus sign, that means this strain is wild type for this gene, methionine and biotin. But threonine, leucine and thymine is mutant gene. That means this methionine and biotin can synthesized on its own, but threonine, leucine and thymine cannot synthesize on its own. When strain A and strain B, when grow in different minimal medium, they cannot grow because of minimal medium lack of this supplement. Strain A have methionine and biotin gene mutant, and strain B have threonine, leucine and thymine gene mutant. That's why strain A and strain B cannot grow in separate minimal medium. But when strain A and strain B mix with them on a minimal medium together, this mixed culture gives rise to some prototrophic colonies, that is methionine plus, biotin plus, threonine plus, leucine plus and thymine plus. The mixing was a genetic cross that produced recombinant, and these colonies are recombinant that produced by the exchanging genetic material between the strain. To confirm this, Bernard Davis conduct another experiment that provide definitive proof that bacteria require physical contact to exchange genetic material. This scientist placed two strain A and B in a liquid medium on either side of U-tube apparatus. This is separated by a filter with pores that is too small that allow only medium, and then used pressure to move the liquid through the filter, allowing the medium to exchange but not the cell. Then the cell placed in different minimal medium to check for the appearance of prototrophic colonies. No prototrophic colonies appear, leading to the conclusion that genetic material is not released into the medium. Cell to cell contact or physical contact will be required for the genetic exchange. And this experiment indicates that E. coli has the type of mating system called conjugation. So what is conjugation? Conjugation is a process of horizontal gene transfer where genetic material, typically a plasmid, is transferred directly from one bacterium to another bacterium through direct contact, often mediated by pilus. And conjugation process is unidirectional transfer, and in this process one bacteria cell is donor and another is recipient. So what is plasmid in E. coli? This plasmid in E. coli also called F factor or F plasmid. F factor is a special type of plasmid in E. coli and related bacteria that enables cell to transfer genetic material through conjugation. This F factor encodes genes that are responsible for the sex pilus development, and sex pilus is a tube-like structure that connect donor and recipient cell and initiate DNA transfer during conjugation. Here is three different type of cell. One is F minus cell, F plus cell and Hfr cell. So F plus cell contain the F plasmid as an independent circular DNA molecule. They act as a donor. And F minus cell lack of F plasmid. They act as a recipient. And Hfr cell, that is high frequency recombination, have the F plasmid integrated into their chromosome. They transfer chromosomal gene during conjugation. Another cell is F prime factor. So F prime factor result from improper excision of the F plasmid. This F factor integrated into the bacterial chromosome, that is called HFR, and this F factor loop out incorrectly. That include a piece of the bacterial chromosome. And this lac plus gene is the bacterial chromosomal gene. When the F factor improperly or incorrectly loop out, this lac plus gene is excised and that enter into the F plasmid. And when the F plasmid carrying the extra chromosomal DNA along with this cell is called F prime factor. So conjugation is not reproduction, it's DNA transfer between living cells. So how does this DNA handoff actually happens? Let's walk through the process step by step. First is F plus conjugate with F minus. This is the F plus cell and this is the F minus cell. F plus cell carry F factor or F plasmid and F minus cell lack of F plasmid. And the F plus cell form a sex pilus. The pilus attaches to the F minus cell, pulling the two cells close together, and conjugation bridge form between the cell. This is the conjugation bridge, and this bridge is the channel through which DNA will move. F plasmid is first nick and then transfer the plasmid to the recipient cell. With this transfer, a plasmid in the donor cell replicates by the rolling circle replication. Here is the plasmid first transfer one strand and then another strand act as a complementary strand and synthesize the another strand. And the recipient F minus thus become the F plus. Here is no chromosomal gene are transfer. This is the plasmid transfer, only not chromosomal recombination. Another is HFR and F minus. This is the F plus cell and the F factor integrated into the bacterial chromosome by the recombination. This is called HFR cell, and HFR cell contain the F factor, and but F minus cell cannot contain the F plasmid. So conjugation begins at the integrated F factor origin of transfer. Chromosomal gene adjacent to the F factor are transferred into the F minus cell, and transfer is time dependent. The longer the mating and more gene will transfer, usually interrupted before the entire chromosome is transferred. The recipient F minus remain F minus because the full F plasmid rarely completes transfer, but it may incorporate donor chromosomal gene. This produces recombinant F minus with new genetic trait. This system is used for gene mapping. So let's use the HFR and F minus for bacterial gene mapping. So here is the HFR strain and F minus strain. HFR strain contain the threonine, leucine, sodium azide, that is azide, ton, that is bacteriophage T1, lactose, galactose, and antibiotic streptomycin. And F minus also present this gene. But here is a plus sign indicate This strain contains the wild type gene, and there is no plus sign, that means these genes are mutant. And here is R means resistance and the S means sensitive. Resistant means this bacteria can survive and grow in presence of the agent, and the sensitive means the bacteria is inhibited or killed by the agent. It cannot grow when exposed. So here HFR stain, here is AZIR resistant, sodium azide resistant, TONR bacteriophage T1 resistant, and STRase streptomycin sensitive. So HFR strain have AZIR TONR, so resistance of this agent. When this strain grow in the medium with sodium azide and bacteriophage T1, then this strain can grow because of the resistant gene present. But this bacterium cannot grow in this medium that contain the antibiotic streptomycin because of the streptomycin sensitive. So HFR strain cannot survive in the medium that contain the antibiotic streptomycin. Same as F minus strain cannot grow in the medium that contain sodium azide and bacteriophage T1 because of the this gene is sensitive. But this strain can grow in the medium that contain the streptomycin antibiotics because of the this strain is resistance of streptomycin antibiotics. Here HFR strain is phototrophic because HFR strain have threonine plus, leucine plus, lactose plus and gal plus. But F strain is auxotrophic for threonine and leucine. Threonine and leucine is mutant, and lactose and galactose unable to ferment. So here HF for A strain and F minus first conjugate and then gene is transfer. First the threonine and leucine gene are first donor gene to be transferred to the F minus at 8 minutes, threonine and leucine gene, and the two genes are inseparable time wise in a conjugation experiment because they are physically very close to one another. That's why two genes are transferred in a same time. Next gene to be transferred is ZIR are seen at 9 minutes after the start of conjugation and 1 minute after the threonine plus and leucine plus gene enter, and ton R recombinants are seen at 10 minutes. Ton R, this is the ton R gene, at 10 minutes it transfer, followed by lac plus gene recombinants at about 16 minutes and gal plus recombinants at about 25 minutes. This is the map of gene based on the time of entry of the donor gene to the recipient gene. Threonine and leucine before the 10 minutes and AZIR also before the 10 minutes. Then in 10 minutes ton is transferred. Then before 20 minutes lac is transferred and 25 minutes gal plus is transferred. And this is the graph that shows the percentage of HFR genetic markers and the time of appearance of gene in the recipient.